So, kia ora and welcome to my presentation. Indeed, welcome to the beginning of your life. My research career has, for the large part, studied life before birth, fetal life. And it is arguably the most important period of our life. It is, if you think about it, the time at which we take those two cells that were so enthusiastically contributed to our existence one day or night, and we grew an entire human being. I mean, it's an amazing feat of biological engineering. But importantly, it sets the very foundation of who we are physiologically as adults. It sets our architecture. And if we get this wrong, then there are lifelong consequences, assuming that we even make it that far. Now, for many of us, we get through this process entirely okay to come out as the well-rounded adults that we are. But many of our children in Aotearoa, New Zealand, don't have that privilege. Nearly 8% of all of our newborn babies are born preterm, as you can see on the left of the slide. These babies are at significant risk of mortality and morbidity, and they have been a large focus of my research. Some babies die before they're born, stillbirth. Some babies have impaired growth, such as the little twin there. Some babies will suffer adverse events at birth, such as oxygen deprivation, hypoxia, that'll injure organs like the brain. And some babies will have adverse events well before birth, in preterm life, for example, that they will then continue and survive with and be born at normal term. And we know that around 80% of all individuals with cerebral palsy, for example, have adverse events well before birth. Now, to make a difference to all of these children, we need to understand their basic physiology. We need to understand their adaptation to the adverse events. We need to understand the mechanisms of injuries, and that way we can develop treatments and apply them correctly if we have biomarkers that tell us those children at risk, where they are in their illness process. And this is really an explanation of what I've been doing throughout my career. Now, we can gain a lot of information from our clinical studies, but to really robustly test our hypotheses, we need to turn to animal models. And the one that we choose to use is the chronically instrumented fetal sheep. As you know, in New Zealand, we have a few sheep. In this model, we can instrument the fetus with recording devices and study them throughout gestation. And then we can deliver them as newborns, study them as newborns, study them growing up. It's a very powerful physiological model. Sheep in New Zealand have driven some very important findings for obstetric and neonatal medicine, such as the steroids for preterm lung development with Liggins and colleagues, and more recently, therapeutic hypothermia, some of you might know it as brain cooling, for brain injury in newborns, led by Alistair Gunn and his colleagues. In the time that I have, I really want to just talk about three aspects of my research. Getting to know about the preterm and its response to one adverse effect called hypoxia or oxygen deprivation, and to understand the importance of evolving brain injury, and in fact, involving time, and developing biomarkers to detect those at risk. One of the objectives when I first started doing this kind of research on preterm fetuses was really to challenge some dogma around our understanding of the tolerance of preterm, either newborn or fetuses, to adverse events. There was this kind of formula around that prematurity equaled immaturity, which equaled vulnerability, that you are so immature in your organ systems and your neural control of those organs that when you were challenged with hypoxia, you could not defend yourself. Now, if you look at the baby on the left, the preterm newborn, this is a baby who is exquisitely vulnerable. It is out of place and out of time. It was born through adverse events, and it continues to face those adverse challenges because it's not, in fact, ready to be born. Its organs are not ready to function in an exudator environment. The supposition was that this vulnerability that one sees in the preterm newborn must be the same for the preterm fetus, because they are the same creature, but only separated in time by the process of birth. And there were some sheep data around from the 1980s from the Americans that said actually there's some evidence to show that if you look at the graph on the right, this was an experiment looking at preterm fetuses and term fetuses, these are sheep, and looking at the exposure to moderate hypoxia. And if you look At the bottom there with the term fetuses, when they're made hypoxic, they mount what is called a chemoreflexly mediated response to hypoxia. They slow their heart rate down to help protect the heart. And this is considered an example of a mature neural system. Conversely, however, the preterm fetuses showed no response in heart rate or indeed in any other cardiovascular function. And so this hypothesis was out there. But I really had some difficulties with this because we had known for a very long time that immature animals were incredibly tolerant of hypoxia. So here's a report from Robert Boyle and colleagues, Robert Boyle, he of Boyle's pressure law, to the Royal Society in London in 1670. So that's the year, not the page number. And they report the anoxic or hypoxic tolerance of animals of different ages and different sizes to hypoxia using a device called the exhausted receiver, or essentially a vacuum chamber, developed by the amazing engineer Robert Hooke. And they reported that very young animals survived 3 times longer than other animals of a similar size or older animals. And this anoxic tolerance we learned about in the 20th century, in the 1950s and 60s, really reflected the amount of glycogen that you had in your heart. And glycogen can produce energy with the absence of oxygen or no oxygen at all. And newborn animals have a lot of glycogen in their heart, far more than you and I. But actually, and you looked at the graph on the left, colleagues from Oxford University in 1960, this is the work of Heather Shelley, showed us the amount of glycogen you have in your heart doesn't peak at term when you're about to be born, but rather in preterm life. So the fetus that you see before you has more glycogen than at any other time of its life. And it kind of suggests that maybe that umbilical cord that you see gets into trouble more often than not in preterm life, and they need a lot of energy. So we queried whether actually this hypothesis that immaturity was somehow detrimental and placed you at such significant risk, or that there was something different about the metabolism that we really needed to understand in our preterm fetuses. And that maybe that original moderate hypoxic episode that I showed you in the previous graph wasn't enough of a challenge to our preterm fetuses. And so we set up and developed a more severe hypoxic model based on including that umbilical cord that you've just seen. And this graph shows you fundamentally 2 messages. But it doesn't matter what age you are, from term to extremely preterm, you will defend yourself with chemoreflex responses that are cardiovascularly mediated, as you can see here with heart rate, blood pressure, and blood flow, in the same manner, qualitatively the same. So it wasn't that immaturity of nerves played a role in that. It's just that we weren't challenging the metabolic demand for homeostasis. So that's one message of this graph. The other message is that the younger you are, the longer you can keep your heart going because of all that glycogen on board. And here is the paradox. You're so good at responding and defending yourself from hypoxia that you place yourself at risk of injury to all organs, in particular the brain, because you are exposed to profound hypotension and hypoperfusion, low blood pressure and low blood flow. And this paradox suggested that actually that was more of a contributor to injury than this immaturity hypothesis. So armed with this knowledge, we wondered if things would change once the fetuses started to recover. If they could survive, did they then go and quickly die? These 2 slides show you on the left the term fetuses and on the right the preterm fetuses for the first 24 hours of recovery, so perfectly oxygenated again. And we found that our term fetuses were more likely to die than our preterm fetuses because they had so much heart injury, cardiac injury. And if they weren't given pharmacological support, they would go on and die. Where our preterm fetuses, they resuscitated by themselves, they made some adjustments in the first few hours and then just basically got on with it. And we have gone on to develop a model which allows us to look out over time from preterm to term life just to quantify how it is that you could survive with a brain injury and just keep going. On the left, I'm just showing you some brain activity to demonstrate the sort of differences between the groups, just in the way in which the brain develops. And on the right, what we've done is we've gone through and looked at different ages and different timings in the evolving phases of injury to look at what patterns we see and what mechanisms of injury there are, because you need that information to develop treatment strategies. Time is brain. It's time is brain for us as adults if we have a stroke, and it's also the same for newborns and our fetuses. And we've learned that most brain cells don't die during the insult itself, but during recovery. And that gives you an opportunity to get in and protect or repair. And studies of newborn babies, humans and animals, that have looked at magnetic resonance spectroscopy to look at energy in the brain have shown that there are phases of injury. That initially energy restores in a latent phase for a few hours, but then that fails again, just as it did during hypoxia, even though the fetus is receiving or the newborn is receiving oxygen. And it can be upwards of 3 days where you have this energy failure. Now, that is the period of time when most brain cells are going to die. Then energy is restored and new cells can be produced. They go on to try and form neural networks. They have lots of problems we're learning about, and there's actually a lot to learn about in that phase right now. But what we learned from all of this was how to develop neuroprotection strategies around understanding the mechanisms of action. For therapeutic hypothermia, for example, it's important that you start at the very beginning of the latent phase, that green box, and you continue to the end of the blue box. Other treatments that we're looking at, like stem cells, we know we can actually start halfway through the blue box and continue into the yellow box, into the tertiary phase. And so understanding timing is very important. But we didn't actually know this for our preterms. Did they have the same phases? If they did, were they the same timing? And so I worked with engineers in London on developing near-infrared spectroscopy, an optical physics technique, to look at oxygenation in the brain and energy in the brain. This is a method that just allows you to use near-infrared light that's absorbed by oxygen and something called cytochrome oxidase in mitochondria. And cytochrome is part of the respiratory chain inside mitochondria that makes energy. And what we can tell you is that preterm functions exactly like the terms in terms of the phases of injury, and the timing of injury is shown by this figure. So we see a loss in that orange phase, recovery in the green phase, secondary loss, and then a tertiary recovery. And about 7 to 10 days, those guys come back online to being normal oxygenation and energy. So this was really great because then we can have a template for developing therapies for our babies, which we're doing. But there remained a key thing, and that is the biomarker that we needed, because we don't know when time zero is clinically. We know it in the lab, we have our clock, we know exactly when we've given the insult, but babies come to us with lots of injuries, with lots of timings and lots of adverse events going on. And so it's important to look for features that occur in the phases that we understand about, so that we can say clinically, yes, this baby has had this, here it is, it's in this phase, and this is the kind of treatment that we want to apply. And on the left, you can just see some physiological data, and on the bottom, we were measuring EEG, brain activity, and it's pretty flat for amplitude or power. But we noticed that in the latent phase, and only in the latent phase, that something was going on that was very fast. So something that wasn't big, but fast. And when we looked at the raw records, if you look at the right, the latent phase, we found these little microseizures. They're very sharp, fast waves, about 200 milliseconds. And we know that now from our studies that they contribute to injury. Now we know about the mechanisms of their control. We know how to make them worse. We know how to make them better. We've now gone on to look at a little clinical trial in humans to see if they also occur in humans. And you can see on the bottom graph, they do exactly that. And we're just quantifying now whether we can predict from their occurrence whether injury in a baby occurred or not. Our big challenge now going forward is, can we apply the same kinds of techniques and technologies to our human fetuses? We can't obviously instrument them, we can't measure their brain activity or their blood pressure. And we're looking for surrogate measures, taking our physiological knowledge and utilizing the clinical tools that we can have. And one of the things we can easily measure and repeatedly measure is fetal heart rate. And I want to end my little talk here just showing you some new clinical data, some new experimental data, and we hope to get some clinical data, and a new clock that we've had to learn about, and that is circadian rhythms. Circadian biology is very important to our physiology, but we know almost nothing about it in the preterm, or in fetal life, I should say, and certainly not in the preterm. And I have one final slide of data to show you This is hot off the press. So these are heart rate recordings of frequency of the variability of the heart over the longitudinal recordings that we can do in our experimental model. In the black shows you, in fact, circadian rhythms happen. So you've got a day-night cycle going on there. Every blue bar is a nighttime, the white bar is day, showing you there's a lovely rhythm going on there. And the hypoxia animals, I'm hoping you can see that there are some significant changes. We lose rhythmicity in the secondary phase, and there's a lot of suppression. So there's a biomarker for the secondary phase in utero just from external heart rate. And there's a recovery over that 6 to 7 days at the beginning of the tertiary phase. And then we have this reduction of the daytime dip in our heart rate variability, which we think is a very consistent feature, and we hope will become a diagnostic biomarker for clinics. And we're looking at humans now to look at that. As always, this kind of work is a huge team of people. This is our current team, and I want to thank them. And funding agencies, of course, and a particular shout out to the Health Research Council of New Zealand, because they have been an absolute amazing supporters for this kind of research in New Zealand. So thank you for your time, and I hope that you're enjoying your day.